And now it is my pleasure to introduce you to Dr. Tom Jenkins, our featured speaker and world-renowned dairy nutrition researcher and professor from Clemson University. Dr. Jenkins has spent his last 30 years trying to help us all understand important issues in lipid metabolism in dairy cattle. He joins us today to help us understand what happens to lipids as they travel through the rumen of the dairy cow and what the implications are for cow health and performance. Welcome, Dr. Jenkins.
Thank you, Renee. And before addressing today's topic on rumen lipid transformations, I first wanted to start by briefly reviewing a few key points on dietary lipid characteristics. Now, lipid in the diet of dairy cattle is highly complex and diverse, but from a practical standpoint, we only need to focus attention on the fatty acid fraction. Now, it's the most important lipid fraction for several reasons. First of all, it's the component in the lipid that has the high energy that we expect out of the lipid supplements, including fat sources we add to the diet to increase production. But some of the fatty acids are classified as essential because they cannot be synthesized by body tissues, yet they're required as precursors for the manufacture of important metabolic regulators in the body tissues like prostaglandins. And then finally, it's a fatty acid fraction that can have a very significant impact on rumen function and fermentation. Now, two important points to lead up to today's topic. The majority of the fatty acids that are in the feed are bound to glycerol as either simple or compound lipids. Now, a good example of a simple lipid is a triglyceride that is the primary lipid fraction in cereal grains like corn. Compound lipids, a good example, are glycolipids that contain a sugar or carbohydrate, and they are the primary lipid fraction in forages. But in both cases, fatty acids are bound to glycerol. Also, the majority of the fatty acids are unsaturated, which is synonymous with double bonds. If it has one double bond in a fatty acid chain, it's referred to as a monounsaturated fatty acids, and we abbreviate it that way. If there's one or more, or two or more, it's a polyunsaturated fatty acid in the chain. We refer to it as saturated fatty acid. So with that background, what we want to explore today is the answer to these three questions. First of all, what happens to this dietary lipid as it passes from the diet to the intestines and undergoes metabolism by the rumen microorganisms? Second, what are the intermediates and end products from that lipid metabolism in the rumen that will accumulate and eventually find its way into the bloodstream of the animal? And then finally, third, how does lipid transformations in the rumen affect animal performance? What is the relevance or implications? Now, these are the three main pathways or events that occur with lipid metabolism in the rumen that I want to mention, starting with lipolysis. Now, this is carried out by an enzyme called lipase, produced by the microbial population, and its job is to release the fatty acids from the glycerol backbone in both simple and compound lipids. So following its very quick and extensive release, it will result in the accumulation of free fatty acids in rumen contents. We could also refer to this as non-esterified fatty acids. The two are synonymous, simply meaning fatty acids no longer connected to the glycerol backbone. Now, once lipolysis is complete and the fatty acid is released, it is then free to undergo the process of biohydrogenation, which acts to get rid of double bonds. So enzymes produced by the microorganisms act on polyunsaturated fatty acids, monounsaturated fatty acids coming from the diet, and convert them to saturated fatty acids that accumulate in the rumen contents. Now, the last one is microbial lipid synthesis. The microorganisms can use VFAs from carbohydrate fermentation and link them together and build up a long-chain fatty acids that are an integral part of their membrane and their structure so that they can grow, divide, and function normally. A common fatty acid produced in this way is palmitic acid, saturated fatty acid, but you also find smaller amounts of unusual fatty acids such as branched fatty acid that comes from isovaleric and isobutyrate volatile fatty acids that are produced. Now, I'm not going to say more about microbial lipid synthesis. I just wanted to mention it, make sure we were aware of it. So the attention then is on lipolysis and biohydrogenation. Now, I thought the best way to depict the changes that occur from this is to run some lipid through the animal and look how it differs from the time it goes into the rumen until it comes out. So I'm going to do this in a virtual cow using CPM as the simulator. Now, I won't go through all the details of the diet I'm going to feed this cow. It's a typical TMR. This one had 60% forage in it, mostly corn silage and some alfalfa hay. And the model predicted that the cow was going to eat 48 pounds of dry matter per day. So let's look on the intake side. What goes in from this specific diet? The model predicted that the cows were going to eat 892 grams of fatty acids per day. Remember, this is specific for this diet and the fatty acid composition of the ingredients that are in this diet and for this particular cow. But in this instant, 892 grams going in. Very little free fatty acids, typically less than 10%, would be free fatty acids, and the majority is bound to glycerol. Low amounts of saturated fatty acids, only 2 grams of 18O, which is stearic acid, 219 grams of 18-1, which is oleic acid, 440 grams of 18-2, which is linoleic acid. That's the primary fatty acid that is consumed by a cow normally. And then smaller amounts of 18-3, which is linolenic acid. And referring back to our discussion on the first lecture series, the oleic acid is part of the omega-9 family, linoleic is omega-6, and linolenic is omega-3. Now, let's consider how that's going to change in going from the mouth to the small intestine of the animal. What's the outflow out of the rumen? And the model predicts that we'll have 876 grams coming out, somewhat similar to the 892 going in. This is typical. There's not a lot of gain or loss of fatty acids across the rumen. So you come pretty close in seeing the same amount coming out as what's going in. But it was only 10% free fatty acid going in, and now it's going to be greater than 80% free fatty acid coming out of the rumen, and that's because of lipolysis. And the stearic acid, while only 2 grams going in daily, is now going to become 534 grams coming out. Oleic acid goes down from 219 to 30 grams. Linoleic acid from 440 grams to 31. And then linolenic acid from 37 grams to just a few grams, 3 grams in this case. So you can see the extensive increase in free fatty acid from lipolysis and the very extensive drop in unsaturated fatty acids from biohydrogenation that occurs across the rumen. Now, biohydrogenation, focus on it for a second. This is the structure drawn out for linoleic acid, we said is the primary fatty acid normally consumed, which is a C18-2, shows each of the 18 carbons and the 2 double bonds in the chain. The goal of biohydrogenation is to get rid of the double bonds. So we want to go from an 18-2 to an 18-0. Now the reason for this, the reason it evolved is because double bonds in the chain have antimicrobial properties. The double bonds or the unsaturated fatty acid concentration gets too high in the rumen, it will interfere with microbial growth and metabolism. So biohydrogenation evolved to more or less detoxify the plant material. Now, in the first step of biohydrogenation, hydrogens are brought in from very specific and select donors. okay, and added to the carbons with the double bonds, which converts it from a double to a single bond. And then it can progressively go down that chain, bringing additional hydrogens in till all double bonds have disappeared. And we end up with what we refer to as a saturated fatty acids. No double bonds, or every carbon is saturated or holding as much hydrogen as it can hold. Now, the process of biohydrogenation does not proceed as a single step directly from linoleic to stearic. There are several intermediate steps along the way. In the first of these, the double bonds are just moved or rearranged. We start with an 18-2, and we still end up as an 18-2, but we simply move the double bonds around to form a pool of fatty acids called CLAs. And then we can add hydrogens and go down to an 18-1, add more hydrogen, and go down to an 18-0. So in this first step, CLA formation, this is the double bond structure in the linoleic acid that I showed previously. In plants, double bonds are normally never any closer than what they're shown here. They're separated by two single bonds. The other feature of plant fatty acids is notice that the hydrogens are drawn on the same side of the carbon chain, and that's to depict the cis isomer, cis configuration. So plants, for the most part, only make cis and never put the double bonds any closer than this. There are exceptions to that in the plant world, but you don't find it very often in TMR of a dairy cow. Now, in the first step of biohydrogenation, the hydrogens are rearranged so that the double bond that was normally here is now moved to closer together. Now when they were separated by two single bonds in the plant, they're only separated by one single bond, and this is referred to as a CLA or conjugated linoleic acid. Conjugated is a biochemistry term that's been in existence for a long time, refers to double bonds that are this close together. Also notice that the hydrogens in the CLA around one of the bonds is drawn on opposite sides of the carbon chain, which is used to depict a trans isomer or trans configuration. Now, the most common type of CLA that's produced is cis-9 trans 11. That's the one I just showed in the previous slide. Now, each of the bars on here is a summary of studies showing different CLA isomers that have been documented in the rumen contents of cattle. So there's a multitude of CLAs. In fact, we know that 20 or more can be produced. You find many of them there at any given time. Normally, cis-9 trans 11 is the most common. That's the one shown here in highest amounts. Now, there's two important points that I wanted to make about biohydrogenation. The first point is while cis-9 trans 11 may be the most common typically, leading to trans 11 when hydrogen is added, feeding, changing the diet of the animal can change the pathways of these biohydrogenation and lead to different CLAs produced. For instance, low pH in the rumen for any extended period of time has been documented to increase the production of an alternate CLA referred to as a trans 10 cis 12. And that one's been shown to be one of a few that can inhibit lipogenesis in the mammary gland, cause milk-fat depression. So the point here is nutritionists are in control of the direction of this pathway and the type of CLA produced. Create a diet that leads to, for instance, low pH in the rumen for an extended period of time, and you're in effect telling the microorganisms to produce more of this CLA, which has a negative consequence on milk composition, milk components. Now, the second point that I wanted to mention was for the longest time, I thought these intermediates of biohydrogenation were too small to be of any consequence. We know that's not true anymore. These minor CLA intermediates of biohydrogenation can have very major effects on animal performance and human health. For instance, on the human side, biohydrogenation is responsible for red meat and dairy products being higher in saturated fats, which is of concern to the medical community and government nutrition agencies. And debate on that concern carries on today. But biohydrogenation is responsible for cows eating mostly unsaturated fat to their products having mostly saturated fat. And the CLAs have very high biopotency. A good example of the cis9 trans 11 CLA that we mentioned, most common one, is a very potent inhibitor of some sorts of cancer and anti-atherogenic and also known to enhance the immune system. On the animal performance side, biohydrogenation can inhibit the supply of essential fatty acids to the tissue. And this may limit the supply of prostaglandins and have negative effects on some systems like reproductive performance or the immune system. We've already talked about the benefits of biohydrogenation in reducing double bonds, having a positive impact on microbial growth and metabolism and enhanced production in that way. And also the CLAs have effects in the animal. And a good example is the trans 10 cis 12 isomer and a few others that are known to inhibit lipogenesis and cause milk fat depression. So with that background, I wanted to summarize, stop there and summarize, and do it by asking a question that perhaps you can help me fill in the answers. This is a matching question, and let's pick out the best answer on the right that best fits each of the statements on the left. So question number 1, which of the answers on the right is the abbreviation of the major fatty acid consumed by dairy cows? Well, among the 6 answers shown there, 3 are abbreviations of fatty acids. Which one best depicts the fatty acid in the diet consumed by cows? And the answer there would be D. That's a structure for linoleic acid that we've been talking about with its cis-cis double bond configuration. The second one, which is the best answer to depict the process that increases free fatty acids in rumen contents? And the answer there is lipolysis. Third, the abbreviation of the CLA normally produced in the rumen, the answer is B. That's the structure of the CLA with the double bonds closer together and the trans double bond. And then the last one is, which one greatly reduces the supply of essential fatty acids to body tissues? And that answer was biohydrogenation, A. All right, one more. Of the below choices, which is the best substrate for fatty acids, essential fatty acids or saturated? And the answer I would choose would be answer B. That includes linoleic acid and linolenic acids. Now for decades it's been shown that CLAs can come from linoleic acid, although usually only one is shown, cis9 trans 11. Now we know there's many more than that. But no pathways were shown to create CLAs from linolenic. But work that I did here at Clemson, my lab last year, showed that CLAs, and in fact a multitude of them, can come from linolenic. So I'm of the opinion that both of these can make CLAs. The omega-9 fatty acids include oleic, which is an 18-1, and there's no known pathway for how microorganisms in the rumen can add double bonds to the carbon chain. So you can't make CLAs from oleic, and the same is true for saturated fatty acids like stearic, where you can't add double bonds. So with that, I want to thank you very much. I'm going to turn the control back over to Renee, and I think she has time for you to ask a question or two. Thank you.
Thank you, Dr. Jenkins, for those insights into the transformation of lipids in the rumen. It is, as Dr. Jenkins said, it is now time for questions and answers, and it looks like we've got a couple questions that have come in. If you haven't had a chance to type in your questions yet, just look for that box in the lower right-hand corner of your screen, and you can hit send, and we'll read your questions for Dr. Jenkins to address as well. The first question we have is, can you give an example or two of feeding conditions that cause that CLA shift that you mentioned?
Yeah, good question, because it does have very practical applications. And one of them that I mentioned already is low pH. We're still in the process of studying rumen environment and the conditions that cause this shift, but there's well-documented evidence, like I said, that low pH can change the pathways of biohydrogenation and the mixture of those CLAs are produced. But another one is excessive amounts of unsaturated fatty acids, especially polyunsaturated fatty acids, is known to cause it. And for that reason, we need to limit the amount of unsaturated fat that we feed to cows so that it doesn't overly inhibit the microbial population and cause this shift. But another one that we know is excessive grain or starch in the diet, and evidence is suggesting that, especially in the case of real rapidly degradable grain or starch sources. So those are three examples that we have evidence in the literature to support there's been a shift in the types of CLAs produced.
Great, Dr. Tom. We've got another question that came in that I'd like to pass along. And this question is in regards to methods to create bypass fat, bypass EFAs of flax lipid. Do you have any comments in that arena?
Okay. Yeah, it sounds like somebody has an interest in being able to get some omega-3 fatty acids that are in flax past the rumen so that we can supply more to the tissues for supporting reproductive performance in other systems. We're limited in those technologies right now. Of course, you can always feed flax. It does offer some natural rumen protection by the nature of its hard outer seed coat, but that's not foolproof. The animal does chew and create breaks in that seed coat, and we process it, which can cause some changes, which can expose the internal fatty acids to biohydrogenation. Other than that, there are, other than mixing oil seeds together and perhaps pelleting them in some manner in order to provide some better protection for that hard outer seed coat, we're very limited in what we can do with flax or oils for that instance to provide protection. So we're kind of at the mercy for how much the animal's going to chew it and the rumen characteristics, if these oil seeds can get through or not, and actually deliver more essential fatty acids to the tissues.
Great, thanks for that answer. We also have a question in regards to what the upper limit of PUFAs are to avoid having detrimental effects. Yeah.
Yes, that is a topic that we want to get into in more depth in succeeding seminar series. So just very, very briefly, we're all hoping that there's some magic number, a single number that we can use that's all going to apply here and make it easy for us to follow some guideline. I want to, in later seminars, talk to you about a little bit more detail on ration characteristics and the types of, the exact nature of the fatty acids that are in these PUFA fat sources we're using in order to make that decision. So I'll have some equations later on that I use that gives me some guidance on this. But for sure, you know, we're looking at not being able to feed, you know, much more than 6, 7, 8% total fat in the diet of dairy cattle before we're going to run the risk of getting into problems. But how it breaks down in PUFAs and other fat sources depends on a number of ration characteristics, and I hope you'll stay tuned for succeeding lecture series so that we can discuss that topic in more detail.
Great, Dr. Jenkins, thank you. I think we have time for one more question before we're up with our 10 minutes of Q&A. And that question is, if essential fatty acids are limiting from biohydrogenation, can this be overcome simply by feeding more fat?
Yeah, that kind of goes back to the flaxseed question that we discussed earlier. And feeding more essential fatty acids from flaxseed or cottonseed or corn oil or soybean oil. Most of it does undergo biohydrogenation and then limit the supply, but some does get through. And the more you feed, the more gets through. But the problem is it's the risk of inhibiting rumen function. And you don't want to cross that line. All right. So feeding more just for the sake of getting more essential fatty acids may be counterproductive if you're feeding so much as to inhibit cellulose digestion in the rumen or limit intake on the animal or adversely affect milk components. So we have to, again, find that line where we don't feed too much polyunsaturated fatty acids. This cross that line relates to the last question. And once again, we want to try to, today's was more background information, but we want to try to pull these practical aspects together here in succeeding lectures. So once again, I hope you'll stick with us as we take this basic information, pull it together, and start to talk about the practical implications.
Dr. Jenkins, it looks like we've got one more question that came through that I think will take just half a minute, if you wouldn't mind taking one more. And that question is, what should an omega-6 to omega-3 ratio be?
Oh, okay. Yeah. Well, in the diet, of course, that's it's usually skewed very, very much towards omega-6 to omega-3, very low amounts of omega-3. But what's important is what comes out of the rumen and what we get to the body tissues. And in human diets, that's very much overloaded on the omega-6 side, and there's the big push that should be more in balance, and a push to get us more omega-3 in the delivered. What we need in these cows is we need to get as close as we can in humans and cows, for that matter. The tissues are all being guided based by the same basic biology, but closer to 1-to-1 ratio is ultimately the goal here. And for cows, it's very much influenced by or limited by the process of biohydrogenation that you don't have to deal with in non-ruminants. But that's a great question, and we can pinpoint that number and try to give it a little bit more attention and detail later on as well. Great question.
Great. Well, it's time to wrap up this segment of the Fatty Acid Forum on the Transformation of Lipids with Dr. Tom Jenkins. We really appreciate you joining us and for your interest in sharpening your ear. This webinar is being recorded, so it'll be available at virtusnutrition.com by Monday at the latest. And then I also want to make sure you mark your calendars for the third webinar entitled Fat Feeding Strategies to Maximize Returns. And that is scheduled for Thursday, September 13th.